in the chat. I see Frank the Tank here, Pet Time. Let's see who else is in here. Squidiot. Oh, interesting name. I like it. Karina. And let's see. Yeah, those are the ones I see. There were quite a few comments, so thank you for being active in the chat even before everything started. Appreciate that. And welcome to the stream. So let's see if I can catch up. I probably will miss some of the things that are in the chat because they tend to go fast. But Squidiot, excited to see some Expansus. Excellent. And these are not just any Expansus. These are Porcellio Expansus Autumnal Equinox, which is a strain that was isolated by Laura at Smugbug. Sorry, it took me a second. Oh, well, thank you, Pet Time, and nice to see you back here in the chat. Maybe you can hear, maybe we can see, this is a baby Expansus right here, but let's get a closer look. The colony's kind of exploded with babies. And as you can see, the color varies quite a bit on these. Some of them have more or less pigment, but they have that sort of caramely, orangey, tanny, whatever color you want to call it. I see Frosty Inverts is here as well. Excellent. And let's take a look. You cannot see, you can sort of start to see the color on these that looks a little bit like an Expansus. Like this one that's in the molting process right there, you can see it a little bit. That one almost looks like a negative version where the dark where the light is in the normal one. Kind of fun. Let's take a look under some of these other hides and see what we can see. Ooh, there's a bunch with some nice adults. There's a nice male. And loads of babies there. Oh, here's Jonathan Romero as well. And this is Karina's first time catching a stream. Yeah, that name didn't seem familiar, and I welcome you. Thank you for being here. That is a really nice male. Check out the color on that one. Would you call that more orange or more caramel? Oh, Wendy Hickson's joining us from the UK. A very hot UK. What are the temperatures there right now? So male and female, Pet Time, to answer your question, there are a couple ways to tell. The easiest way to tell in this species and in some other species is these exopodites on the Europods, these two long tail-like appendages. Technically, they are the exopodite Europods. Isopods typically have, at least the terrestrial ones we're familiar with, have 4 Europods. And the exopodites are the long ones that you can see clearly. In a few species, you can see between those, you can see the endopodite Europods. But anyway, the males have long exopodite Europods in this species and in several others. And then here is a female with much shorter exopodite Europods. There are other ways to tell by looking at their pleopods, but it's a little bit more complex because you have to turn them upside down, or you have to put them in a clear container and look from underneath, and you have to have pretty good eyesight. So this species is much easier to work with, and the others that are like them. Many of the larger Porcellios have this trait, the longer exopodite Europods. Sirena, welcome. They look kind of blonde, yeah, that makes sense. They do look kind of blonde, don't they? And this male, even though it's not the biggest male I've ever seen, it's a pretty decent sized male and looks pretty cool. And Jonathan, I absolutely love that you found this channel too. There's a whole bunch of wee ones. So Sirena, hard to say. In my experience, they're not particularly seasonal, but I think it depends on a lot of factors. I think it may depend on the temperatures they're kept at and various other things. So for me, they don't seem to be that seasonal. I've seen babies at any time of the year, but there are a lot of isopods that reproduce a lot more in the spring. There's a female with another crop of babies here. And I see Filippa from Denmark. Welcome. And there's Warren. Thank you for joining us as well, Warren. Nice to see you back in the live. Let's see, was this the one I just had, or is this a different one? I don't know. Every one that I pick up seems to have loads of them in it. So this colony is really thriving. This colony started in January 18th of this year. So I've only had it, 6-ish months, and they're doing really well. They're actually doing better than my original Porcellio expansus colony right now. I mean, it's not doing terribly badly, but I'm seeing a lot more breeding in here than I am in my other one. I'm not quite sure why, because the other one bred like crazy when I first got it, and I moved it up to a bigger bin with partially new substrate and stuff, so I'd expect them to go wild, but they haven't done as well as these. Squidiot, this channel has inspired me to start the path of entomology. Love that. I love that. Every time I hear something like that, somebody's inspired to basically do anything that furthers the hobby, whether it's a career choice or they've started to work with a new species, anything like that, I love to hear it. So 33 Celsius is still pretty warm, isn't it? So temperatures like 102 Fahrenheit, that's about what we're starting to deal with now where I am too. Now, is that your house temperature or is that the outdoor temperature? I'm praying for you that that's not your indoor house temperature, that's just the external temperature. Because if your house is 102, we got some issues. One thing I would say though, you can put small containers, you can go to the store and get those reusable ice cubes. Basically, they're like a plastic encased or silicone encased ice cube that you put in your freezer. You get a bunch of those, and then you can put one in the corner of your cool area, your hydration station in your Isopod bin, and then just one in one corner, and then that becomes a cool area that they can go to and make sure that spot is also moist, and the condensation will help make it moister. That can help if it's really, really hot. You can do that. Don't put too many because you don't want them to freeze, and some isopods are not particularly cold sensitive, and that won't bug them at all. Tropical ones, if they get too cold, of course they can die. And of course freezing solid, that can be a problem for any isopod. But what I'm saying is you don't need to overdo it. But something like that you can then just throw back in your freezer and cycle some in and out. When it gets really, really hot, that can be a way to help you deal with the issues. And Karina, but you're right though. They are stunning, but so are any. You're right. Sorry, I'm just having you all stare at egg crate. It's terrible. I gotta do better than that. So, what other species would you like to look at, everybody? What I'm doing right now, thought I'd do this during the stream. I'm checking on the moss, checking on the leaves, see if they need a little refresh, because that's one of the jobs of my kids is to check on those things and let me know how they're doing so I can refresh them if I need to. This one doesn't look too bad in terms of the leaves, but I'm going to throw some in anyway because it never hurts. And if you have isopod requests from the species that I have, I'll do my best to honor it. Okay, so Newburger, just found grindleworm or some kind of whiteworm eating the food in your trichorhinatomentosis container. So as far as explaining why that happens, it's common for the substrate of isopods to have enchytraeid worms like grindleworms or whiteworms in it if it's not pre-sanitized, and there are benefits and drawbacks to pre-sanitizing substrate. One of them, the drawbacks, is that you get, you're destroying the microbial life that's beneficial for the colony, and you're going to have to figure out a way to get that going again. And if you don't have it going and you put isopods in stark enclosures, there's a break-in period that's it's a little harsh on the isopods, to be honest. So one of the advantages of sanitizing that substrate is that you don't have issues like that happen. Enchytraeid worms are not particularly problematic for your isopod enclosures. They will compete with them for food to some degree. Other than that, not a big deal. If you have fish or small amphibians, small terrestrial amphibians, or even small aquatic amphibians, those enchytraeid worms, whether they're grindleworms or whiteworms, are going to be an excellent food. Excellent food for a lot of those creatures, and you'll do better. You'll do great just to use them. So if you have something like that, go ahead. If not, you can try starting out with new substrate if you want to get rid of them, but it's not really going to cause a huge problem. Okay, Karina would like to see some maculatum or cubaris. I think we can make both of those wishes come true. Let me move some stuff really quick, and I'll try to answer some other questions as we keep going. If I miss something, don't feel shy about asking again, because I'm not going to be able to catch everything. So if you have another question you'd like me to answer, then go ahead, and I will I will do what I can. Let me see. Right here, I'm going to show you this microvarium. This is from microvarium.com. They sent me one of these to try out. And there's some zebras. These are Armadillidium maculatum, just the normal striped morph in here, as well as experimentally, there's an Oreo crumbles, Porcellionides pruinosus, running around like crazy. A fairly large one because it grew up in here without other members of its own species, so it's diverting all its resources to growth rather than reproduction. And there's a milkback in here as well. There's a whole bunch of zebras of various generations. I do have thousands of zebras in another enclosure, but there goes a milkback. Basically, I tried to make a community of different isopods to see what would happen as an experiment in here. Just wanted to see what happened. And basically what happened is some of the other species died off. I had a rubber ducky in here. I had an Armadillidium nazatum in here. I had a Porcellio ornatus nord in here. The only three species that survived were the zebras, which reproduced, the Porcellionides pruinosus and the Porcellio laevis milkback that I showed you. I thought perhaps that the zebras were hybrids at first, possibly, because I saw an Armadillidium klugi that I had in here mating with the zebra, and I wasn't sure when I first saw the babies what was going to happen, but it turned out they don't seem to have crossbred at all because there are multiple generations in here and they all look exactly like zebras. So what must have happened is, even though that zebra was very, very tiny when I put it in, must have made it already at a surprisingly tiny size, because these are all descended from that one individual. I do like these microvariums, by the way. They turn out, the substrate's great, the decor comes in great, the instructions are great, the container's great as long as you use it for a very small group of isopods. You know, I'm going to have to move these up to a different enclosure after a while because, you know, zebras are going to keep breeding and they're going to overtake this enclosure. But for now, it's working great. I want to show you some more isopods here. I'm going to grab some cubaris. How's that? I think that's what Karina asked for. So here we go. Some cubaris. Well, this first one I'm going to show you is, I think, been reclassified to Nezodilo, but then I have some true cubaris for you. Well, we don't even know if they're true cubaris. Currently classified as cubaris, let's put it that way. But I do love these. These are the red tigers. Nezodilo red tigers. Part of the reason I love them is because of the variety. If you look at the differences in these, this one right here is more of a shiny sort of look to it, while the others are more matte. Also, the patterns vary quite a bit, so they're very cool. One thing too is they tend to really, really congregate on bark. Not that isopods, you know, in general isopods do that. There's one that looks like it's working on molting right there. Let's see if there's some under this piece. Yep, there's one. Ooh, they do tend to scatter when irritated. See, that one just fell down. Cute little things though, with the typical face of so many isopods. Fun rostrum there. Fun species, pretty easy to keep. It's a little bit more prolific than the rubber duckies, and they tend to make their own little spots that they come back to on the bark, which is fun, like their own little custom hideouts. Let's see. And I'm just looking at the chat. Oh, Warren wants to see the Jestroys. We'll totally do that. And we can do Japanese versus American Magic Potions. Sure. Black Hoffmannseggi. Yeah, we could do that. Oh, Frosty Inverts bought your first tarantula from Bugs in Cyberspace. That is awesome. Peter's great. Jesse's great. They're amazing people. And they will totally help you out. They've got great stuff, great knowledge. That's very cool. Here's some Panda Kings. You can see a little tiny Panda King right there, and there's another small one right there. They're breeding. This is a newer colony for me, fairly new, but they're still breeding pretty well. They're one of the faster breeders of. You can see a bunch of them there down there in the substrate. Here's some more of different ages and sizes. If you want a cubaris that breeds pretty well, Panda Kings are probably one of the ones to do. So, Sirena, your Porcellio not as high yellow crashed. Oh, I'm so sorry to hear that. Well, did the whole colony, you lost all of them? Porcellio ornatus high yellow have a lot more yellow markings on them. We can actually take a look at them in a minute, as opposed to yellow dots. Yellow dots tend to be a little hardier, and maybe the yellow dots need a little bit more moisture. That's I'm still sort of up in the air about it. It kind of depends on who you talk to and whatnot, but that may or may not be the case. Okay, I'm going to grab some Jester, just a second. Okay, Warren, these are for you. It was your request, wasn't it? The Jester. This is absolutely one of the best Armadillidium species. Now I'm kind of scared a little bit, but they're all over this enclosure, just crawling everywhere all the time. Love it. And Warren was mentioning that he loves his Jester too, so I love to share this species. Ava bees, there are actually lots of terrestrial isopods that can't interbreed. In most of the cases, I would say, if you're looking at the same species, they're going to interbreed, but there are some cases where we may have misidentification of species, cryptic species, things going on, so that they're not going to interbreed, but in general, if they're the same species, they can. Porcellio laevis is kind of a taxonomic mess right now. There are morphs that don't interbreed with each other, and that may be because they're different species, something may be going on there. But generally, if they're not the same species, they won't interbreed, but I have heard of some exceptions. It looks like the Porcellio unites in the hobby probably have some hybridization going on between Floria and Pruinosis, for example. I believe Oren McMonagle had some Armadillidium versicolor, I think it was, crossed with some Klugii, and successfully produced those. So it can happen in some cases, but generally doesn't. If real pandas could breed that fast, that would be true. So Squidiot, to know what I've heard about Spiky isopods. Very little, to be honest. I have not had any experience with them myself, not a bit of it, and so I don't know what to tell you there. I need to get somebody, look at all these baby Jester though, just check it out. Flood of baby Jester, love it. The Spiky isopods, I need to get an interview. Somebody out there knows somebody with spikies that would be interested in doing an interview with me. I'd be all over that. So let me know if you have them or somebody else has them and want to do an interview with me. I know some people who do and might want to do an interview, and I can check in with that too. But if you happen to know, I'll be there. Okay. One thing I love about the baby Jester is that they already have the yellow markings on them. You can see almost immediately, and they're quite tiny. They still have the yellow markings. It's so cool. And they don't lose them. They don't become more dim with age. They're just super cool. Let's see. Good beginner isopod. Well, the zebras I was showing you a little while ago, definitely a good beginner isopod. These Jester are a good beginner isopod. I would say little milkbacks or another type of laevis are, as long as you're prepared for some intense breeding. Armadillidium vulgare, another big one. Porcellio scaber is a good one. So Karina, I don't actually have Bolivari or however you say it. I don't have that one. One of the few giant Spanish Porcellio that I have never kept. Oh, Frank to think I love Zingers too. I do have some, and they're breeding. I love the Zingers. Got those from Brandy at Ractastic Racks. Be right back with some more isopods. I don't remember who asked, but somebody was asking about this species, Porcellio Hoffmannseggi black. Another species that is dimorphic. Males are much more elongate and they have the longer exopod at uropods than the females. So let's see what we can find here. A lot of these are immatures that I'm seeing right now. Let's see if I can find a mature male. This one, the big one right in the middle of the screen, is obviously a female from those Isopods. Just have to dig around. There's a young male right there. You can see it's got the longer Isopods, the Exopoda Isopods. Let's see. There's another male right in the middle of the screen. Can't tell what that one is because it's covered up. Probably a female. Yep. Need some more mature males. Where are they? Bunch of juveniles right there. Different sizes. Huh. And they're certainly producing plenty, so there must have been males in here at some point. What's going on? I mean, they can mate when they're younger, so maybe it's that. I don't know. Those little ones have weird coloration right now. They probably change over time, but huh. Are all the males just fleeing, or just don't have any fully mature males in here right now? Maybe that's what's going on. Tell something about Armadillidium nazatum orange. Okay, I've got some of those. I can show those. And I also, it's going to take me a second, so I'm going to leave these here so you can check these out while I grab some other isopods because it's boring to look at my shelf. Okay. All right, switching these out. I just saw a comment, somebody said, what's the worst beginner isopod? Well, I think that depends, but I would say I would not recommend Porcellio magnificus as a beginner isopod, nor would I recommend necessarily rubber duckies, because if you spend $200 or $150 or something on rubber duckies and find out isopods aren't your thing, it's kind of a big investment to find that out. These are yellow dots. Porcellio ornatus yellow dot. You can see most of them have some yellow dots on the posterior end of their peryon and on the pleon, and they're kind of irregular. They're not super flashy, but they get fairly large and robust. They're very active. So those are yellow dots. Not super colorful, but still cool. Okay, now we're going to look at the high yellow. Get an idea how they differ visually. Their care is very similar. I think the high yellow might be a little bit more tolerant of dryness, possibly. If so, it's still, isopods don't want to dry out ever. There's none there, just springtails. Let's see. There's got to be some around here somewhere. Probably mostly in the, there's one. Probably mostly in the cardboard stuff. Let's take a peek over there. Yep, there's a ton of them right there. See, these are the high yellows. I find them a little bit more picky, a little bit less hardy than the yellow dots in general. There's a couple more. Color is absolutely gorgeous on those. A little bit more picky. Perhaps a little more dryness tolerant. As they grow, they get more yellow. You can see the younger ones are a little bit sort of not as bright as the bigger ones. And Warren says he's enjoying the Isopod zoology book. Oh yes, the one by Warren McMonigal. It is a great, great book. Glad you're enjoying that. Grab a few more isopods here. What was I supposed to be grabbing? Can't remember. if I forgot your request, just let me know. I'm happy to, do what I can while we're still running. See what I can do. I think most of Porcellio ornatus are seasonal breeders to some degree. They tend to just have a burst of breeding in the spring, and they'll sometimes breed at other times, but not as much. These are orange Armadillidium nazatum. They're a little paler in color and a little more of a true orange than the peaches. There's a bunch of them under here. You get more of an idea. This piece was closer to the moist hide. And Armadillidium nazatum, they do tend to like their moisture. So you can see you've got some color variety in here, but most of them are pretty orange, especially when you get close up and look at them. Some of them are darker, like that one that's just in the middle right there. But that is the orange. Very similar to the peach in many ways, but not quite the same. JH, you didn't end up keeping them because they breed too much, huh? Yeah, if you keep isopods, you breed isopods. That seems to be the truth in most cases. Somebody made a request for, I think it was Armadillidium vulgare Japanese magic potion, and let's see, Japanese magic potion and American magic potion. Okay, this is my Japanese magic potion bin. Let's take a peek. I am going to just propose, and I've started to reflect this on my website. The magic potion really should only apply to the American line, because the reason why it's named magic potion is because it was line bred for more yellow markings. Now, what is often called Japanese magic potion is a beautiful isopod, as you can see. I mean, they're gorgeous. But they were not line bred for the yellow markings, so they do have some yellow markings on them. But they're basically a pied Armadillidium vulgare. That's basically what they are. You see they have some yellow markings, they have some darker markings. Some individuals have more dark markings, like this one right in the middle of the screen. More dark markings than many of the others. The older ones tend to have more, but not always. These don't get as large as the American magic potion either, so I think we should call these Japanese pied. That's my proposal. Can we call them Japanese pied for that reason? Armadillidium vulgare Japanese pied. That is my request. Can we do that? It helps disambiguate the two lines, and hopefully helps people from crossing them, because that would be bad. One thing about the American line is they get so much bigger. The individuals get bigger, and we could lose these traits like that if we're not careful by just haphazardly crossbreeding. You know what I'm saying? They're the same species, but they're definitely different lines, different strains, and we want to keep that integrity. So these are the Japanese ones. Right here are the American, the true magic potion. You can see a lot of them have a really lot of yellow. It's very intense, and it's more widely distributed on the body. They're just overall more yellow. They get bigger, so they are quite a bit different. And it's important to maintain the integrity of those two lines. You just get the overall impression. I mean, look at this. This looks like I took a highlighter marker to that isopod right there, because of this line breeding that has occurred with that in mind. So we really ought to keep the strains disambiguated with the verbiage that we use, so that we also keep the lines separated and unique. Because there's nothing wrong with Japanese magic potions. They're awesome, but they're not the same thing. So we shouldn't call them the same thing. And Squidiot, the care is 100% identical. I do not care for them in any, there's no difference, because they're exactly the same species, just different lines. So their care is the same. And this is a strain that is a mixture of, oops, sorry, now we're looking at empty containers, and there's my other bins. Okay. So, oh yeah, Frank, I would never want to drop one, that's for sure. This strain, also sometimes called Magic Potion, is more properly called Orange Dalmatian, best called Orange Dalmatian, because these are descended from Magic Potion, crossed with Orange Vigor. So you do see some of the increased yellow, but many of them have the rusty color, and I think not all of these do. Some of these just have the dark ones, and they may have more Magic Potion blood than they do Orange Dalmatian, but some of these in this enclosure have sort of a rusty red color from that from the orange ones that show up in their ancestry. Orange Vigor crossed with the American Line Magic Potion. Sorry, that is, the light saturation is messing up how that looks, but these are gorgeous too. Probably one of the more underrated ones out there. And overpopulation Sirena definitely can make a culture crash, just because of, well, the ecological concept of overshoot. If you get to the point where the population has exceeded the carrying capacity of the ecosystem or the microcosm that we've created that attempts to mimic an ecosystem, basically if you run out of any of the resources they need, then the population can start dying off in mass because there's not enough of something, whether it's space or food or whatever. And Newburger. I don't have any spiny species. The best I've got is bumpy. I've got some tuberculated species, like Armadillidium peraci, but that's it. No spikies, although I think that would be fantastic. So I'm going to grab some random species, but if you have something that you would like me to take out, I will be happy to do it. Somebody was asking about the Zingers, or they mentioned the Zingers. So let's take a look. There's a Zinger right there. These appear to be, and it depends on who you talk to, but there may be some wild localities that look like this. Someone was telling me, I think it was Scott Maroney was mentioning this, that there are some wild localities that have this pattern, or this color. They're just black and white, but these were isolated. Brandy from Ractastic Racks isolated these from her yellows, and they were true breeding. And she kindly just sent me some, which is fantastic. There's some more adults right there, and there's a bunch of little babies crawling around doing their thing, being axanthic zingers, which, I mean, most of the time, to be honest, if I see an axanthic something, I don't think it's an improvement. And I'm not going to say this is an improvement over the striking yellow of Armadillidium gesteri, but I don't think it detracts from it. I think they're just as gorgeous as the wild type gesteri that they're descended from, which is strange for me. Most of these xanthics are not like that, but these are, for me, just to be honest. And Squidia, I think the name does have to do with it. Oh yeah, Jackson, yeah, thank you for your question on Patreon, and now I guess you've seen the answer, so I should check Patreon and see if there are any more questions on Patreon. Jackson was asking about diving beetles, just if anybody had kept any, and I have not kept any. I think I need to at some point because they seem fantastic, especially the sunburst ones that Peter at Bugs in Cyberspace will sometimes have in stock. Absolutely gorgeous. Okay, Sireno would love to see the papayas. Okay, I gotta warn you that my papaya colony is new. So they're breeding, but it's a tiny colony still. I haven't had it that long. So let me dig it up here. There it is. I've got the glacier, Cubaris marina glacier, and Cubaris marina wild type, and the papayas. Pretty cool. One cool thing about them is that they have been approved because they do exist natively in the United States. It was pretty easy to get them approved, all the Cubaris marinas. I have seen Pseudo-armadillo spinosus. They are incredible. I don't have any, but I think they're very cool. Yeah, Jackson, Peter has a pretty good video on the care of those guys. Oh, look, it's my pie dish beetle. I keep my pie dish beetle with my Cubaris marina papaya, just because the care is very similar. Peter sent me this critter, and it's fascinating. The appearance of how its body is shaped. It's got this concave body structure. I hope you can see that. I don't know if it's showing up, but it's pretty cool. See how that looks? Whoa! Just dropped. Poor little bugger. Yeah, Peter sent me this quite a while ago, and it's just fantastic. Does excellently well. It thrives apparently with the Isopods. I've had it in here for a good long time now. I think he shipped it to me in, I want to say October. Is that right? Of 2021. It's just been doing well with the Isopods. Walking water dish. Love it. I think that's his new name, Walking Water Dish. And Newberger, you're welcome. I'm glad that I helped you learn a little bit about bioluminescent insects. There are actually a lot of bioluminescent creatures that we need to know more about. Okay, there's a little group of papaya right there. Very cool. Like I said, it's a new colony, and there are babies in here. But they're kind of unique because they're pink. There aren't really very many isopods that are pink. I don't think there are any that are pinker than these Cubaris marina. They have those lovely little faces. Don't get large. I don't know if you can see down in the substrate, you can see some babies kicking around in there. So they're breeding for me. They're pretty decent breeders. Cubaris marina probably breed faster than many of the other species that may or may not be Cubaris that have the name Cubaris. Walking upside down bottle cap. Yeah, that's a pie dish beetle, Pitts fam. So Frank the Tank, superworm beetles, I've heard of them lasting at least 2 years. 3 years sounds pretty good. To be honest, I think superworm beetles are underrated as pets because they're amazing, they're easy to produce, and they're very active during the day, out during the thing, much more active than a lot of other beetles during the day. So if you want the day active beetle, not great for handling, not super bad for handling either, but they will emit that stink if they're upset. So let's see, what else am I digging up here? Trying to find my isopods. Here. This one's fun. Yeah, the smell of those beetles is the only downside to them. Cubaris marina are super easy. Just keep them like any other isopod, really. Just on the moist side, make sure they have plenty of moist side. Uh-oh, it looks like somebody dumped food, or maybe that's just slime mold, I don't know. I have to check. I hope nobody dumped food on that side. There's some Paracae, the bumpiest isopods that I have, the closest to Spiky that I have, so not very close. Probably one of the most prolific armadillidium species out there. So Cubaris marina is super easy. Keep the moist side quite moist. Maybe at least a third of the enclosure can be on the moist side, maybe half even. And they're very, they're not super picky on anything else really. Some airflow, doesn't have to be a ton. They're really pretty easy. So Frosty Inverts, the pie dish beetle, came from Peter at Bugs in Cyberspace. I had an expo coming up and I said, Peter, can you help me out? I need some good assortment of beetles for this expo because I have some, but I want a few more different sizes and shapes for this expo, just for a display table. I was doing an educational table. And he said, sure, and he hooked me up. He and Jesse hooked me up and sent me some beetles, including that pie dish beetle. So I brought the pie dish beetle in a separate enclosure because they're not really desert beetles. And then the desert beetles I brought in another enclosure, and then we had a lot of fun teaching people about them. It was cool. So thank you again, Peter and Jesse, for doing that. So I do keep dart frogs. Right now I only have one species. I have the bumblebees. They're actually not in this room. I did a feeding live stream of them not too long ago. But I should do another one so you can check out my my dart frogs. They're Leucomelas, the bumblebees. Sorry, Catherines. I actually did this partly for you and people like you in other time zones so that you could catch it. It'll be easier to catch it. So we got about 20 more minutes, which is kind of fun. So Zach, you have the same kind of dart frogs I have and the same number of them. Oh, Jackson, you're going to be in your new place soon. Awesome. And Mr. and Mrs. Morelia. Sorry, the stream was a little weird today in terms of timing. Sorry about that. Summer is always a little crazy, and I bounce around a little bit on the stream, I'm afraid, on the timing. But I'm glad you're here. I'm glad you made it. Let's see. This is what I showed on my Instagram recently. These guys. I don't remember if that was today or not. I just put a snake shed in here. Let's see if there's any under here. Oh yeah, plenty. Silisticus convexus U crane pied. One of the most underrated isopod species in my opinion. Love the way the pattern expresses because you get everything from almost completely white to almost dark, but everything in between, and most of them are pretty high expression. They're also extremely effective as a bioactive cleanup crew, not likely to cause issues. There's a chunk of snake shed right there still. They don't like the belly scoots so much. As you can see, that's all belly scoots. But they are basically bulletproof isopods, not likely to cause a problem in a vivarium. Love them. And yes, I'm glad that you managed to catch some, Catherine. And how your mantids doing? And squidiot. Yeah, I think it depends on where you are about the wild-caught velvet ants. As far as I know, there isn't regulation, like federal regulation on wild-caught velvet ants at all, because they're not a plant pest. So I don't think they are. I don't think there are any problems particularly there, but there might be local issues in your state or whatever, so it's good to check in on that. So Johan Exotic, hey, welcome. Suggestion for an isopod for a dart frog vivarium with high humidity. Well, of course, the dwarf whites work well for that. You can also do something like Silisticus convexus. They do really well in high humidity as well as lower humidity. And you can do something like Atlantoscia floridana, the Florida fast isopod. I'm going to check and see. Oh, here's some of my armadillo officinalis. These guys get big. They're pretty cool. So those are some of my suggestions. Nagurus cristatus is another one, and Isopodus species tarragona. Those are like my 5 go-tos for that situation. You could also do dwarf purples. Yeah, I love velvet ants, Squidiot, as you may know if you've seen my videos on them. I have had quite a few velvet ants over the years. Love them. I try to keep a few every year or two, get them more because they're gorgeous. You know, my daughter saw a velvet ant not too long ago. I was hiking. With family when was it? It was last month, I guess. And my daughter saw a velvet ant, and I didn't hear her say she had seen it until it was too late because it had gotten away by the time I heard my other daughter say that it was there. Okay, I'm kind of just taking what I can grab here. Oh, okay, Squidiot, I see. So that's how you got into it. Cool. And Rutger, well, welcome and congratulations on your new isopods. Fantastic. Those are 2 good species to start with. Just be prepared for them both breeding a lot. So I wanted to see if we could see any yellow zebras. These are just so fantastic. There's one. Should we see if we can scoop that one up and take a peek at it? Yellow zebra. Look at that. That one's almost more spotted than striped, but still gorgeous, right? I have some that are more striped and some that are more spotted in here. There are babies in here too. There's a different one. There's a whole bunch gathered right there. I don't know if you can see them. Well, not a whole bunch. Now that they're taken off. But there's some. Some running around. And Ava bees. I am in the state of Utah. There are plenty of velvet ants here, but the variety increases, the variety in species increases when you get into the southwest part of the state, where you can actually find the white velvet ants like Dasimutilla gloriosa, one of my very favorites. Oh, Warren and Dr. Plants. Yeah, zebras are awesome. Can't beat zebras. One of the best beginner isopods, but nobody should be without them. Let me grab some more here. What are we going to look at now? Oh, I don't know how many of you have seen Armadillidium nazatum pearl. Cool species, underrated. Well, it's a cool species, but it's a cool morph, is what I meant to say. They occasionally have little flecks of color on them, but they're mostly this kind of pearly color, and it's really cool. Oh, Squidiot, you're in Utah too. Cool. Yes, Clint's reptile room is only about an hour and 15 minutes away from my house. So it's not too difficult to get there. So you can see the one right in the middle has some flecks on it. That's not terribly common, but they do occasionally have those flecks. There's another one right in the middle of the screen with some flecks on it. Most of them are just kind of the pearly color, and they're pretty cool. And yeah, it does. It certainly makes collaboration easier when you live in the same state, not super far away. The fact that both have a lot of common interest in creatures helps a lot too. Yeti isopods, I love it, Warren. Now, that's particularly apropos coming from you. I love that. I want to show you the. Whoa! I want to show you my moss. No, I don't actually. That was an accident. My camera just bounced. Okay. What I wanted to show you, actually, was these. These are the whiteouts. So they're similar to the pearls, but unlike the pearls, they have no actual pigment. Even their eyes are white. They're just basically snow white isopods, as far as actual pigment goes. Pretty gorgeous. These could be Yeti isopods too. I'm not sure which one wins. Oh, Jackson, Porcellio Scabbard Party Mix. Highly recommended. One of my favorites. I know I have a lot of favorites, but they have a lot to recommend them, for sure. Very cool. Let me get one of my party mix enclosures out really quick, if that's okay. Let's see. I'm grabbing some other isopods too while I'm over there. Might as well, right? Okay, I think this is the right one. I'm gonna have to play around for a second. Okay, so party mix. Let's take a look at party mix. Okay. What can we see? I'm trying to make it so you can see them better. Let's try that. Okay. Party mixes, there's so much color and variety going on, because you've got the powder blues, you've got the oranges, you've got whiteouts, you've got Oreo crumbles, you've got orange crumbles, and then you've got some weird things in between. And there's some calicos and some caramels that seem to show up in this enclosure too. So just about any combination you can imagine shows up in the party mix. There's some more. You can see an orange crumble right in the middle of the screen. There's an Oreo crumble, there's an orange, there's some powder blues. There's, I think that's an orange crumble, maybe a young one. So many different things going on in here. This is my original culture of party mix right here. Let's see what we can see in here. I think we might actually get a better look at them if I can get the camera to do what I want to do. See that one, that was a high white Oreo crumbles that was in there. Where's a good piece of substrate? There's a ton of them right there. There we go. You can see a couple of caramels in there, I think. Some of the whiteouts. There's one of the fading ones. I'm not even sure what to call those. They show up in here. Pretty cool. Something that descends from this isopod culture, but is not part of it. Something I want to show you next. The reason I started my party mix is right here. I wanted to produce orange crumbles. I originally purchased orange crumbles from Kyle Candelian at Roach Crossing. He isolated them a long time ago. And so I started mixing to make my own. I did buy some from him, but I ended up buying some from him. And then the one I got from him were doing really, really well, and then they got too dry and the colony crashed, which is so bad. But right here, I've been isolating, and you can see, I'm still working on it. There's still a lot of other isopods in there, but there's a very, very high incidence of Oreo crumbles in here. There's the occasional dark Oreo, and there are some oranges, but a lot of these are orange crumbles. I've been working on that for a while. Still culling these out. I need to take this colony and split them really well and get as many of the other morphs out of there as possible, because there are enough orange crumbles in here now that I should just split them, separate them out. They're very, very cool. Very, very cool effect. Let me see if I can zoom in on those a little bit. You can see what's going on. So anything that's in there that's not an Oreo crumble is probably het for the other gene. So the oranges are probably het for crumble, and the dark crumbles are probably het for orange. So there's probably a lot of that going on. So Catherine, I'm gonna have to try that. I've never tried to go fishing for scabers with a banana string. That's a great idea. I gotta try that. Oh, the Crystals, Pets, and Plants is here now. And Splatterjack? Armadillidium tyrolense. Ooh, I have not even seen those, I don't think. Sounds so cool. Party mix or gem mix, you mean in terms of the name, or in terms of how gem mix is usually Armadillidium vulgare and party mix is usually Porcellionides prunosus? I don't know. They're both pretty awesome. I love all the mixes, to be honest. Anything that's a mix like that, I love it. Sorry, I'm just trying to make sure I can see the chat. Yeah, I'm hoping Mr. and Mrs. Morelia, you'll be the first to get some of these. In fact, if you still are interested in doing that, I'll make sure that that is the case. Once I get them fully isolated, I'll make sure you're the first ones, because you were the really first ones to express interest in them. And so, yeah, we can make that happen. Oh, Johann Exotic, did you get all the ones that I mentioned? Okay, dwarf whites, Nigerus cristatus, Silisticus convexus, Atlantoscia floridana. Oh, and Isopod species Tarragona was the last one. So we've got about 5 minutes left, Aftech. Okay, cool. Then yes, Mr. and Mrs. Morelia will make that happen. You've been a great supporter of the channel, and I'd love to make sure you get first crack. I'm going to make it a point to go through this and cull out the other ones and put them in with my party mix, so that all I have here is orange crumbles, and I'll probably still have to play with that a little bit because of hets and whatnot, but we'll get there soon. And Pitsfam, a lot of the Armadillidium will eat plants. That's a good thing to think about. Newberger, what's the oldest isopod culture you have? Have you ever changed the substrate? Oh yeah, I try to change the substrate every 6 months to a year. I don't always get there, but I try to. And the oldest isopod cultures, I should say, because I got them at the same time, dwarf whites and powder, no, not powder blues. Dwarf whites, Porcellio scaber orange, and Costa Rican purples. The Trichorhina, what is it? Biocellata, I think. Let's see. And I've changed the substrate in there loads of times, but in fact, the enclosure, there's probably nothing in that enclosure now that was in the original enclosure, and it's probably not even the original enclosure, to be honest. But here's my Porcellio scaber orange. You can take a peek at that. And that one looks like it's in definite need of some new leaves. Let's take a look. Let's see what we can see. Porcellio scaber orange. Falling all over the place. Oh, I'm too zoomed in. You didn't get to see the full effect of that. How about this? There's a bunch of little babies on there. So yeah, this substrate's been changed out quite a bit, so it's not really representative. I'm going to go ahead and dump some leaves in there because it obviously needs some more leaves. If there aren't a lot of leaves in your isopod enclosure, you should probably think about making a change. That's kind of how I think it. So pro tip on changing substrate, if I can, I split it. I split the substrate, rather split the enclosures, and I can't always do that because I'd run out of space real fast. But if I can just split the enclosure and do half the substrate in one, half the substrate in another, and then put new substrate in the other half, you're golden. The other way to do it is just take out 3/4 to half of the substrate somewhere in there, and sift through that, take out the isopods you find, then put that old substrate in a new enclosure or a different enclosure, and water one corner and put a hide in that same corner, and then pull out isopods every couple weeks for a couple months until you're pretty sure it's empty. If you want to be really thorough, that's how you can do that. Well, it looks like we're just about done, folks. Can dwarf species coexist and live with top dwellers without outcompeting them? It seems like it can happen sometimes, but it has so much to do with the particular setup, has so much to do with whether or not there are abundant resources, how much they end up actually competing as opposed to how much we think they should be competing, that kind of stuff. So it can happen, it can work. The bigger and more varied the ecosystem in the vivarium and the more different the species are, if you have one that really is a surface dweller as opposed to a burrower, that'll help too. So it can be done, not necessarily the easiest thing to do, but it certainly can be done. This right here is my one and only Marulinella species. They're very fast, and you can see some babies in there. These are Marulinella species Vietnam, which may not actually be Marulinella, but they're currently considered Marulinella. And right now you can see springtails and baby isopods zipping around. There's an adult up there in the corner, ran under the food dish, I think, somewhere in there. There it is. They're fast little critters, aren't they? You can see the orange on it. Kind of fun. These were a gift, and was at the last expo. They're fun. I'm interested in the fact that they run extremely fast, and they seem to spend almost all their time under the substrate. But yet they have an interesting color going on. Well, I think it is about time for me to wrap up, but thank you everyone for joining in. And Felipe, I'm glad you were able to watch, and the rest of you who were able to watch, like Catherine, who usually doesn't get to watch because it's late. I wish I could deliver to Canada. I can't. I don't have the permits to ship to Canada right now. But yes, a like would be appreciated, everyone, if you're able to do that. And keep your eyes open for my next video on Friday. And thank you for joining in the stream, and I hope to see you next time.